Today, we have again, I used to use the term dynamic because the persons are like that only in Mysore University. They are doing excellently. They are well with academic as well as research activities. So therefore, I am using that prefix dynamic professor with me today, Professor H.B. Manjunath. Professor H.B. Manjunath has qualified both Masters and PhD from Bangalore University and served for a couple of years in Karnataka University, Dharwad, Sericulture Department. He has also contributed for the growth and development of Sericulture Department in Karnataka University. Then he shifted to Mysore University as a professor and he is presently working as chairman. More than 32 years of both teaching as well as research he has. And he has been awarded so many prestigious fellowships like Commonwealth Fellowship. Under this fellowship, he had been to UK, Cambridge, and worked in Wellcome Trust Sanger Institute. And 2 times he had been to Biology Center, Czech Republic, under INSA ASCR Scientists Exchange Fellowship Program, and where he worked for 2 years. Then Professor Manjunath has contributed richly to the field of entomology in general and sericulture in particular. Of course, he has contributed a lot of new species. Just to quote few, he has identified 4 new species of Spingidae family members in collaboration with the Canadian Center for DNA Barcoding, University of Gulf, Ontario, Canada, and Czech Republic Spingidae Museum. He has also contributed wiki species and developed a mobile application called Siri app for sericulture farmers. He has evolved 6 new bivoltane breeds and their hybrids. They based on the heat shock response principle. He has also developed a concept of research on silkworm thermal biology, developed new silkworm breeds. And also he has developed a concept of research called Siri Bioinformatics. And he's also developed a model system for evaluation of medicinal compounds in India using silkworm as a model organism. And also developed technique to use silk biomaterials for therapeutic applications. So both the field-oriented and laboratory-oriented research, he has contributed a lot. And he has made a lot of memorandum of understandings with so many prestigious institutions like Czech Republic Spingidae Museum, Karnataka Innovation and Technology Society, and Accurate Additive Labs Private Limited, where the silk, fabrion, and sericin used for medical and dental applications. He has also made collaborative research with Laboratory of Molecular Cytogenetics, ASCR Institute of Entomology, again Republic of Czechoslovakia. He is a member of so many professional bodies like Associate Member for International Sericulture Commission, France, American Society for Cell Biology, USA, Korean Entomology Society, Korea, Entomological Society of America, USA. Even in India also, for so many professional bodies, he became a member. And he is having a rich experience and doing a lot of work with academic associations in various universities, both in Mysore University and outside Karnataka as well as in India. About more than 5 research projects, major research projects worth rupees more than 50 lakhs completed, and presently 4 research projects are going on. And he has guided more than 7 PhD students, working currently 6 PhD students for their PhD, and more than 65 papers he has published in prestigious journals, and he has deposited so many DNA as well as protein sequences in sequence data banking in NCBS, and also he has involved with various academic assignments, as I said previously, in the university, outside university. with this brief profile. I welcome you, sir, on behalf of myself, on behalf of Academic College, and on behalf of Department of Geology, University of Mysore, and on behalf of all the participants to this session. Thank you, sir. Over to you, sir.
Yeah, thank you, sir. Good morning, everyone. First, I apologize for the delay in my presentation because although I've been using the same system, the desktop, it's more today it is causing some disturbances. I'm sorry for it. Hope you hearing me?
Yes, sir. Yes, sir.
Okay.
Exactly.
So, kindly excuse me for these technical interruptions in delay in my presentation. Anyway, I take this opportunity to wish you all happy Shivratri. Let God bless all of us to take care of health and wealth and our academic endeavors. And I'm able to state that, especially today is the first day for me as well, because so far I've been working with HRDC technology as the coordinator of these UGCs, given a brief introduction about it. And thanks to him, as he knows that I've been working with proteins is long, but I never imagined that I'm going to convert it as a technology. So this is my first lecture with respect to the Silkworm technology, and especially starting on the auspicious day of the Shivratri as well. So all of you know very well that any living organism in Earth respond to heat stress. That's what we measure in terms of heat response. Accordingly, we are going to test it with respect to biological function. So let us have a look into this, how a cell function in a biological system. All of you know that. So we are having mass of cells. Each and cells or every cells grouped into tissues and they perform different functions in our biological system. Collectively, we have a good biological system. So it is all working around the central dogma of molecular biology. As you all know that, all this information is stored in DNA. Then, as you know, it is giving information through RNA to form a protein. Ultimately, the protein is the one which is going to be destructive to us. So when we say about it, all these biological function taking place in our system, and we differentiate it as, with respect to DNA, we say genome and genomics. Then with respect to RNA, we say it is a transcriptome and transcriptomics. The proteome referring to the study of the proteins. Put together, for all these studies, we are going to use another discipline is bioinformatics, where the data of the genome, transcriptome, proteome can also be used. Then eventually we can able to derive a functional unit of it. So, as I recently surprised to see that human has nearly 30,000 genes which is assigned on the side. Then nearly 90,000 genes are well predicted or analyzed, but it is coding for only about 40,000 proteins, which is measuring about 33%. Surprisingly, when I look into the Drosophila has the Drosophila has 16,000 genes in the genome, but it is coding for 22,000 genes, so 22,000 proteins. It is so surprising to me. Earlier, we used to believe that one gene coding for one protein, but now, is it true or no? I think it is not, because a gene can code for too many proteins that can be modified through modifications. So when I look into this Mysore again, there are 18,000 genes as I understand. As I know, the Silkworm genome sequence is already published, but the amount of protein is identified is 9,000, only less than 50%. So it is a question for me, how this differentiation does occur. Of course, this is the one which is making me to transfer whatever the principle and hypothesis of heat shock response we have been studying from bacteria to human into a novel technology, so which I call it as heat shock technology, and this is what I'm going to present you now. Towards this, especially, can you see the slides properly?
The slides are not moving, sir.
Slides are not moving?
No.
No, sir, not moving, and also it is half only we can see, not complete slide.
Is down?
No, sir, not yet.
Is it now? Okay.
No.
No, sir.
No, sir.
Now it's okay.
Now it's
I'll-
moving.
I'll-
Sir.
yeah,
But you
I'll-
reduce the size of the screen.
Yeah, because the full screen is not visible to you, right? See now?
Only half of it visible, sir.
Now it is full screen is visible to you?
Make it full screen, sir.
You can reduce the slide size so that you can see the full slide.
Yes.
That 100% you can reduce this to 90% so that you can see the full slide.
A little bit. Yes, sir. Yes.
It's okay, sir. It's okay.
Now it's okay.
This is okay?
Yes, sir. Yes, sir.
Yes,
Okay,
sir.
can the slide is moving?
Yes.
Yes, sir.
Okay, thank you. So then I thought of whatever the studies I have made so far, then with respect to the principle and hypothesis of heat shock response, where these responses are come from bacteria to human, then one by day, of course, I'm going to tell you where this navigation does occur. To understand this especially, first of all, we need to know what is exactly we are going to have. So, with respect to the heat shock response, the first physiological function, as we have seen, it's defined as ability of an organism, as we understand, to acquire resistance to the fluctuating environmental conditions, even with respect to heat stress after a period of particular exposure. So these organisms are exposed to high temperature or a critical temperature or a threshold temperature. They do have some response, which is measured as heat shock response. So this is eventually is referred as evolutionarily conserved, both in terms of genomics as well as the function. But it is going to be vary with respect to different organisms, as I see. In this context, the heat shock response is support the physiological adaptability of organism against stress, therefore it could be able to overcome. So as the studies is taken place, eventually, substantially, then we come to know that the greater the initial heat dose. So that means if you increase the heat shock temperature, then an organism acquire a different magnitude, then it could able to express this resistance, which in terms of we call it as a tolerance for a longer period. So these expressions of tolerance due to heat shock might last for 3 to 5 days or several hours. This is with respect to true with all the organisms. But in case of silkworm, so we find it much difficulty because it is negatively correlated with respect to the synthesis of heat shock protein, with against the synthesis of silk protein. So then, when we say thermotolerant silkworm strands, they have a tolerance wherein it is synthesizing heat shock proteins, but it is eventually affecting the synthesis of silk protein. So therefore, it becomes a big task for us how to make use of this heat shock response to improve the tolerance as well as the productivity. Then we looked into this particular point. Why not we increase the heat shock temperature in the beginning? Therefore, whether it can increase its magnitude in terms of the genomic tolerance level, then it could be able to survive for a longer period. So as a result, then we start looking at the various aspects. Though it is a ubiquitous, we say, but it varies from not only the different species, but also within the population as well. Then we start looking at the temperature at which HSP synthesis induced, and how long it is going to be survived, and how many types of HSPs are synthesized, where all these HSP collectively involved in cell tolerance, affecting on the survival of the organism with respect to the productivity. Then we start looking at different development stages, especially in case of silkworm, we could be able to see egg stage, larval stage, pupa stage, and adult stage. In this context, whether the egg stage is good, whether the larval stage is good, or pupa stage is good, adult stage is. Of course, the substantial experiments as well as the results are available in this regard. I'm not going to explain in detail about it because I'm just focusing on briefly what does this means to transfer into the technology. So then, the question is, do we have all this information in our hand to go ahead about it? Whether that information which is in your hand with respect to an organism, if you're working in a different organism, you can also look into that. Give you substantial information. But as I understand in Silkworm, there's a big vacuum. So
Excuse
then,
me, sir.
yes, please.
Slides are not moving, sir.
Really? Now?
No, sir. We are in 5th slide only, sir.
So you can put the presentation. Yeah, it's not moving.
But I'm not going to use the animation. It's difficult.
Sir, please click on slideshow, sir.
I think it's not moving.
Yes, sir.
Is it moving?
No, sir.
Is okay?
Sir, make it to full screen, sir.
Yeah, I made it a full screen only.
No, sir. Only normal slides, sir.
Not moving?
No, sir.
How about others?
No, sir. If it is in this,
moving,
there is
sir.
some problem.
It's going.
If it is there, if you don't put in presentation mode, sir, let it be this mode, then you can move the slides.
If I go to this mode, then it's difficult to show all the content because there is animation something.
Now it's moving, sir.
Yeah, it is moving with on the slide
But
mode.
if it in such case, you have to close this and again open the file, like again open the PPT.
I'll go for new share. Let me see. I'll go for new share. Otherwise. I think with the new share is okay. Is okay now?
Sir,
Either you have to share the entire screen or once the full screen is done, after that you can share the full screen.
So the slides are moving now?
No, sir. So okay, so let us go with this. I think it's taking time.
Okay? No.
sir, no problem, sir. If it is not, if it does like that, I just scroll the bar. That's all, sir. That's okay.
Yes, sir. Now it's good.
Fine?
Now it's okay. Yeah, fine.
Fine,
Now
sir. Fine, sir.
yeah, it's great.
Yeah, thank you.
It's moving, sir. Full screen.
Yeah, thank you.
Okay.
Thank you.
Thank you, sir.
Yeah. So, sorry for some technical issues here. So then we started off looking at the different organisms. So it's a question now whether we have all information to take it further or not. Then we understand in Silkworm it is still difficult. We have a big vacuum about it. So then we started exploring all this response in Silkworm to not only understand what is exactly, but also to go further how we can able to develop. So this is exactly what I say, that the beta-protein work is not started recently, but has been started in the 1962 itself. With respect to the Drosophila, it is done by Richardson. Then the term beta-protein was identified coined by Zir in the 1974. It has been working with mammalian cells, yeast cells, Livelihood, Drosophila, mouse. Keeping this background in mind, so then I could able to understand whether all these supporting the Hitchcock response with respect to tolerance as well as other biological parameters. But since Drosophila does not produce any kind of protein in terms of hormones or musca or Luciopodina or human, then it was a big task for us how this Hitchcock protein synthesis as well as sensitive protein synthesis affecting each other that remain enigmatic in Silkworm. So therefore we started exploring. In case of Silkworm, it has been started in the year 1987 by using the cell line, but the work has not much progressed for a long time. Then in 1994, once we completed into open up the paper in short response that has identified some amount of attraction. Then in 2006, especially, we made a first attempt to correlate Hitchcock response, Hitchcock protein expression, and the couple parameters. Eventually, now in 2020, we could able to fix it up the Hitchcock protein expression. If this is a smaller quantity, it is beneficial. If this is a larger quantity, how it is going to work. That we just link with the advertising, where excess amount of Hitchcock protein, if it is synthesized, then it is going to affect on the biological system. So having all this information in mind, then why don't we apply this Hitchcock response to produce new Silkworm breeds, which can be useful for the farmers. Then you know that especially we could able to turn out a good piece of work to by using Hitchcock response, converting it into Hitchcock technology to produce new Silkworm breeds which are in field. So these are the constraints we come across in the event of our research, especially if you say Hitchcock response, it is not going to be a same or uncommon in all the organisms. Then it should be clearly delineate, which you could not able to understand it before unless there is a query. Then we have to understand first what is Hitchcock and what is going to affect our biological system, and where this acclimation comes, and then hardening of organism. Probably in plant system, you could be able to see all this, and how it is going to be useful in Silkworm is very difficult. So we start correlating that with respect to the genes and the stress proteins, how we are going to relate it with each other. As I did question already and mentioned, so though we are studying on Hitchcock protein gene expression, but all the biological and commercial traits is affecting. So then we started looking towards how this Hitchcock response can utilize in Silkworm to acquire thermotolerance and then the new bits. For that, basically we went for the first step is protein basis of analysis. So we started studying the expression of Hitchcock proteins. So these Hitchcock proteins are known as chaperones. Why we call them as chaperones? Because they are helping other proteins to protect their food and assume their proper functions. It acts as an insulator. That means once Hitchcock protein is expressed, that is going to protect the segregation as well as the degradation of normal proteins, which are essential for growth and development of an organism. This is happening in a cell. So we call them cells are vigilant. They are highly vigilant. Then they look for its proper functioning by getting the right fold of this protein. If there is a misfold, then it is going to affect on the normal life. Therefore, in any organism exposed to higher temperature, the normal proteins are the one which is much affected. If their normal fold is not going to substantially support the functional aspect of a cell or organism, then those organisms either they survive or they die. So there we came to know, that if a change is small, then it is good. So when it is going to be deadly, if it is higher, then in Silkworm, we come across this change, whether it's good or deadly, become complex. Therefore, we say that this phenomenon in Silkworm is enigmatic because it needs a lot of information. Toward this end, we started using the Silkworm edge. Then we expose the Silkworm edge to the heat shock temperature of 30, 35, 40, and 45 degrees centigrade. It's a different temperature we use it. So here, when the Silkworm eggs are exposed to the lower temperature, it is going to grow and hatch. But where these Silkworm embryos or Silkworm eggs are exposed to high temperature, the Silkworm embryos are grow, but the larvae, at the time of the hatching, they die. And we say that the 45 degrees centigrade is lethal. So then we start focusing on only the 35, 40, 45 degrees centigrade, ignoring 40 degrees centigrade, ignoring 45 degrees centigrade. But after some time, this the temperature has given a tremendous results and response to us. So then we extended this work to the larvae, wherein we expose the larvae, first instar, second instar, third instar, fourth instar larvae, as we see that. The first, second, third instar larvae solubility is less, whereas in case of fourth and fifth instar solubility is more. Then it is very surprised for us, though it is a larvae, but how different instar is going to respond. Then we started looking at the protein profile through gel electrophoresis. We could be able to see expression of 90 kDa protein in first instar. Then subsequently in fifth instar, we could be able to see more number of hits of protein expression. So we could be able to identify 84 kDa protein, 62 kDa protein, 60, 47, 40, and 33 kDa. So in case of early instar, there is one or two, either HSP 70 or HSP 90 was expressing, whereas in case of later instar, large quantum of expression is appearing. Then it was a big question for us. Why the earlier stage express only one or two instar protein? Is it sufficient for them to grow, show the protein growth and development? In case of 15 star, why it is more? Then we could be able to know that all these instar proteins collectively work together to increase the resistance to the Silkworm larvae. Then it's a question for us. Does it allow us to identify a protein of protein interest conformity through gel electrophoresis? With that, we have to answer yes or no. For that, we will for Western blot. Whatever the proteins are expressed in gel electrophoresis, we need to be get confirmed by immunoblot, for which we subjected that into Western blot by using monoclonal antibody. So HSP90 protein expression even in the normal larva was there to maintain homeostasis, especially in the fluctuated environmental conditions. Whereas in case of the larva which is exposed to 40 degrees centigrade, it could be able to express more protein. So therefore, we could be able to say the hits of protein 90 expressed in all the 5 instars consistently and even consistently in all the instars. Then we could be able to say yes, HSP90 is required for larval growth and development in all the instars. So this is one story. The next, when we come from, when you study this in case of 15 star, this hits of protein expression, though noticed in all the instars, but in case of male and female, so that was a surprise whether they are sex specific or not. Then therefore, we start differentiating the male and female larvae and isolate the protein separately, run the gel. When it's subjected to the Western blot, we could be able to see the difference between male and female. The Western blot clearly indicates that the expression of HSP90 and 70 is sex specific, where HSP90 was expressing more in female and then less in male, whereas HSP70 was expressing male, less in female. So this is what we came to know. As the hits of protein expression in all the instar as well as it is sex specific. Further, we extended this work to quantification by using qPCR, that is a real-time PCR, you know that. Then you could able to see when you have a different Silkworm strands. So qPCR says that HSP90 though ubiquitous, and as you know, it is there from bacteria to human, But within the larval population, within the strands, CSR2, MB42, dizo, and pure mice. You see, comparatively, the pure mice are known for resistant to disease, and CSR2 known for less disease, known for susceptible to diseases, but productivity is more. And this is the negative correlation for the scientists feel. It was important for us. Then, if there is a resistivity, then how we could able to extend the productivity? If there is a productivity, how we could able to go for increasing the resistivity? So therefore, scientists start using pure mice to test with the CSR2. But that is solve the problem at the level, but the cocoon character is not completely developed. So therefore, then we will start looking at with the articles the different male and female. Then it clearly indicates that the female HSP90 is expressing female and the male is. So then we thought of using and defining female and male based on HSP expression separately. So this is what we started working out by using a sex-limited Silkworm strain itself. So with this electrophoretic, and then what we inferred is both HSP90 and 70 genes expressed in all the genes, all the instars, male and female larvae. They are consistently and conjointly expressing, so therefore they are working together. Most importantly here, as we observed, these first instar, second instar, third instar larvae, where HSP is expressing, it is preferential. It responds to the thermos test. So then, when large amount of HSP is expressing, then we started looking at the silkworm larvae where it is expressing higher proteins. When we expose the silkworm larvae to 45 degrees centigrade, though they die, but there is a large quantum of protein expressions. So then we started looking at, though which of proteins are expressing in a larger quantity, but why the larvae is dying. So then when we went for a different interactions, of course, one of the reviewers suggested that I think there is some phenomena is going on in the body of the silkworm larvae. Therefore, we looked at it. So then it becomes, the change is good as you understand, but sometimes it is deadly because it is complex. So whatever the HSP proteins are expressing in the larger quantity in the body of the silkworm larvae, if it is going to degrade very soon, the larvae survive and go over to the normal life. Otherwise, they die there. So therefore, the larger quantity of extra proteins expressed will remain in the body of the organism than that leads to the death. So that's what we publish in a paper. Probably, if you want more detailed information, you can refer to Manjunath, which is published in Archaeology in Separate Results in 2020. So, in spite of the massive production of HSPs due to heat shock at 45 degrees centigrade, all the larvae were found dead, which is a strong proof of concept of autophagy. So therefore, now we are linked with autophagy. If anybody was interested in autophagy, please look into this aspect and get substantial information and derive some kind of hypothesis of phenomena. So how this autophagy is going to be affected? Either it's a stress or whatever, especially when a cell is exposed to the heat stress. In normal condition, you could be able to get the normal folding. But if it is not going to be that, then proteins are degrading. In that case, higher accumulation of certain proteins induces physiological imbalance. So that's what is happening in case of 45 degrees centigrade when the silkworm larvae is exposed. So therefore, it became a bottleneck problem for us how to proceed further to exploit the histoprotein expression in terms of building new silkworm breeds. So then it is defined as a danger signal for a cell or an organism. So as the single dimensional electrophoresis information is not substantial, then we extended to two-dimensional electrophoresis. Eventually, you could be able to find the expression of different proteins. If you see here, there are three spots. These are the three spots, and where there is a small spot which is observed in the normal larvae. Upon heat shock, then you could be able to, when you compare each other with these three bands as a landmark, three spots as a landmark, and this small spot as our point of interest, then it is overexpressed. Eventually here, if you see, So there is no substantial protein spots here in the control larvae, which are subjected eventually the 2D. Then, in the sample extracted from each individual, we could able to see 3 spots. So these 3 spots are identified as isoforms of HSP70. And now we could able to come to know that HSP is not alone, but there are isoforms as well. Then we started looking at comparing the HSP90 is the one, and in case of single band appear in the SGS pause, when it is confirmed with Western blot as well, then we could able to confirm it to dimensional electrophoresis. So when these spots are appear in two-dimensional electrophoresis, then we need to identify what the protein spots are. Therefore, we could able to see the difference here. So here, please have a look of the circle, then see the look of the circle. So here we would able to see only 1 spot, but here we would able to see the 3 spots which are appear. Then we focused on that, what exactly. Eventually, there is another circle which depicts, as I explained, that there is 1 spot which is missing, of course not expressed, but here we were able to see the another spot. So that made us to go further. Then we started comparing all this, how they are. So we mentioned as spot 1 identified as HSP68, the spot 2 identified as HSP71, then spot 3 is identified as HSP70 isoforms. So this is how all the 3 spots are identified as HSP70 isoforms. Subsequently, we were able to identify other HSPs and the HSP90. So then the question was, though we identified the protein by mass spectrometry and all, but how to assign a function to it? By the time, of course, we subjected this paper for publications, but one of the reviewers said, why don't you go for as a function? Then we developed one protocol to make use of this mass spectra and then identify the location of each gene, that is automated gene mapping. That is what we established by using the tool. Eventually, we were able to see, so the protein spots what we identified localized on chromosome 9. This is how we could able to identify HSP70 is localized on chromosome 9. So the single dimensional electrophoresis and 2-dimensional electrophoresis given. In addition to that, we could able to identify some specific protein as well. So in another study, we could able to identify vascular ATP synthesis upon nature, elongation factor 1 alpha, Rab-like protein alpha, and then all these proteins are essentially required for proper functioning of HSP90 as well as HSP70, without which probably HSP expression, if you study alone, is not going to be useful. So therefore, we make it as a complex phenomena. This is also, we made a localization on the chromosomes. Let's see that. So, so for this functioning of HSP9070, as we estimated, it required at least 1 ATPase cycle for proper functioning of HSP9070. In the final complex, HSP90 required ATP bound state. To this, upregulation vascular ATP synthesis is also required. On this, making HSP90 and 70 work together to draw a biological function in an effective manner to acquire thermotolerance. So whereas elongation factor of Bombex-Mori also upregulated with the heat shock, wherein it promotes the GTP-dependent binding of amino acyl tRNA. If anybody interested in this, please look into that, how the GTP-dependent is going to be helpful for protein biosynthesis. So with all these studies, we come to the conclusion that Bombex-Mori responds to everyday fluctuating thermal condition upon expression of each protein. It is not a simple phenomenon, mere upregulation of the genes, but it is also supported by, as I said, with other stress proteins. Then, with the protein informations, then we started working on using biotools to understand much better. For this, what we did, whatever the structure signature what we acquired, then we use metric science, wherein we could able to identify the protein based on spectral signature. So what we did is, whatever the spectral signature we got it, we just pasted here. The moment we pasted these MZ values, then when you interestingly search, if it give you hit results, then it is related to protein which is already there in the database. If it is not, it is going to be new protein and its signature. Eventually, we could able to get that HSP spots what we identified is HSP70 and its spectral signature as well as the peptide bands. Then we could able to identify the similarity, homology, and then sequence string. So we started using the sequence string for further application where we could able to identify the primers and then we can able to amplify the gene. If the gene is present, we can easily amplify the genome. This is some open avenue for us. So after getting this HSP90 informations with respect to the Bombitz-Mori, then we started comparing with other insects as well by using the cluster omega. Of course, all of you know that. Then we pasted the amino acid sequences here. Then we would be able to get the multiple sequence, pairwise sequence, and multiple sequence, however you know that. But interestingly, so when you compare the Bombitz-Mori HSP70 Drosophila, always we say that ubiquitous, but you could see this entering the sequence is not matched. Similarly, there is some gap. Then we started identifying the work has been done with the Drosophila, may not be possible with the Silkworm. Then separately, we start looking at Silkworm alone because it is having the only 70% homology, remaining 30% is not homology. But in another literature, it says that only one amino acid sequence variations is there, then it is available in specific case. So therefore, we started looking at where is the big gap which is present in the sequence similarity as well. And that is how we could be able to say that isoforms of a biological significance may also differ by presence or absence of a single amino acid. If you were to study the Hitch proteins or any protein, even a single amino acid is going to be, if it is varied, then it's going to have a different biological functions. So HSPs that are expressed to the Bombitz-Mori may not find a match in the Silkworm genome database. So therefore, we could be able to identify, we could be able to develop a new bioinformatics tool. So we started using mass spectral signature to identify different. Then we started annotating them. Towards functional proteomics, we started using the proteomic data, and then we use both the dry lab and wet lab procedure. And the concept was developed is protein gene localization. To identify the gene on the genome, we generally use the DNA, but here we use the protein MZ values. That MZ values, when you use, then peptide match, we got it. All this peptide match, but we obtained with the sequence string, we could able to compare or identify cDNA sequence. When you identify the cDNA sequence, of course, it is came to my, it was suggested by one of the reviewer. I thank the reviewer as well. So now we don't need to construct any cDNA from the tissues for further application with respect to the protein. If you have MZ values, then you can take the MZ values, go to the Silkworm genome databases, and then you just paste it. Ultimately, you're going to get. So this is what the cDNA sequence was placed. Then the output was the localization of the gene on the chromosomes. This is what we call as automated gene mapping. So the HSP90 localized on chromosome 27. Once this automated sequence identification on the genome was done, then we started using that to go for a dry lab, wet lab procedure. At the end, we could able to see that HSP90 and 70 was expressed in first and second instars, whereas in case of 4th instar, HSP84 was expressed, and 5th instar, 70, 62, then log 60, 52, 43, 33, all are expressed, which are correctly work together. Eventually, the higher level of BAM HSP and BAM HSP90 transcript notice in male and female larvae, which we say that it is sex specific. But in case of silkworm or probably in an organism, HSP 90 and 70 genes expression due to thermal stress is not only sex specific, but also we proved that it is preferential in first and second and third instar, and both of them are coordinated with each other, wherein it not only supports the survivability of an organism, but also it supports biosynthetic activity of the silkworm larvae, where it can able to produce the couples. If higher expression of the protein is noticed, that is going to cause an imbalance, so therefore larva dies. So therefore, what we see that if any person is exposed to higher temperature, we call it as a sunstroke. It is all because of this overexpression of its protein. This is what we calculated, and this paper what we have published here. So these are the papers. So what we said then, after identifying all this, then we may start using the serial library information. Then we constructed the primer. That means we have designed the primer by using prime blast. So whatever the primer we have designed based on the proteomic data, but not on the genomic data for your information. So whatever the proteins we identified in single dimensional electrophoresis, western blot, two-dimensional electrophoresis, and then we use the bioinformatics tool. Based on that information, we design the primer. Invariably, we design the primer based on the genomic information to identify the gene. But we are going in a reverse wise. So whatever the primer we designed based on the proteomic data could able to identify the gene in the genome of the Mysore and amplify HSP 90, HSP 70, and HSP 40. So after having that genome PCR amplification, then we started using the backbones which are developed in Japan. So then we started interacting with the Japan scientists. Those who have developed the backbones for symptom genome mapping. Then we started using one marker gene for the chromosome, another marker gene for our gene. Then we started using fluorescence in situ hybridization technique, wherein both the probes are labeled with FITC as well as the Texas red. As a result, we could able to see here. So in case of silkworm, the chromosomes are dot-like structure, but here we started using the packaging chromosomes. When you use the packaging chromosomes, especially they are somewhat longer, therefore we could able to see the localization. We could be able to see the position of the gene. As a result, we labeled the backbones. So when we label the backbones and hybridize and label and hybridize under the chromosome, we could able to get the signals. So we could able to see the signal very clearly that the green the green color, it is backbone labeled with FITC. Then it is giving you the green color representing it is 9K80 marker gene. The red one, the red colored signal is the backbone, which is HSP90. We could able to identify that. Subsequently, we started correlating the dry lab procedure with the wet lab procedure, wherein both of them are synced. So successfully we could able to say the localization HSP90 gene in the chromosome, and then in the metaphase stage itself, so that we could able to correlate with that automated one what we did with MS exact values. So with this, we could able to say that both in case of wet lab procedure and dry lab procedure, we could localize, we could identify HSP90 gene on the chromosomes. So then we started using we sequenced this HSP90, HSP70 each protein as well, and then we correlated the Silkworm genome project, and then we identified the difference what you have seen there with respect to the sequence differences. The pure Mysore, as I said, it is resistant to high temperature, whereas and before it is a biotin. Between polyvoltane and biotin, we could be able to see HSP90 sequence is dissimilar. And this is what created a good interest in us to take it further, how we could be able to develop a new Silkworm. See the differences here. So if you go for some kind of comparative analysis, though it is a Silkworm, but these two are styled, then the HSP expression is not only species specific, strain specific, but also sex specific. Then we started working with designing the different primers to know what is exactly. Then the question is, if a long gap is noticed in the endogame sequence, what is the biological connection between the two sequence strands? It shall be examined with other silkworms. Probably, if anybody is interested, please look into that. We can take up this paper. And these are the sequences which you are uploading. So after having all this information, then we look towards its functional significance. So therefore, we differentiate it, we call it as a functional omics. Then, whatever the information we get so far, then is it possible for us to develop a silkworm breed? Then, to show, if you want to develop a new breed, what is the consequences? We looked at now the need of the hour. The need of the hour is, because of the global warming in the sericulture industry, is silkworm breeds or the strands which can thrive very well in the hot, fluctuating environmental conditions. So then, we could be able to say that whether they are totally tolerant, if they are totally tolerant, what about their cocoon productivity? Then our interest, we don't want to sacrifice the cocoon productivity, but only thing, we need to know the transition phase of this total tolerance, wherein the silkworm larvae should overcome shortly with the fluctuated environmental conditions. For that, we came to know there is a phenomenon which is called as acquired tolerance. Then, instead of going for total tolerance, we started looking towards acquired tolerance. To acquire tolerance, what are you supposed to do? For that, then we need to make an organism to acquire tolerance at a particular point of time, and then it should come to the normal. So therefore, we could be able to switch over the expression of the synthesis of feedstock protein in a particular time, then it can stop the synthesis of feedstock protein, but the same machinery can switch over for synthesis of silk protein. And this is the concept what got into my mind. Then the question is how to improve this quality and the yield through acquired tolerance. So, is it possible to have a silkworm strain or silkworm breed or any breed which is thermotolerant with good productivity? Then the question is how to achieve this. When the question is that, then it requires sound knowledge on silkworm thermobiology. Otherwise, it's not possible. Whatever we need to know, we should have information on feedstock. That's what I already explained to you that. And acclimation, how it is going to do that, and how we achieve. So we have established the basic knowledge, or whatever the basic knowledge we have acquired so far. Then we started integrating all these informations with proteomics, genomics, transcription, bioinformatics to evolve a new breeding strategy, which is called as a develop proteomics, genomics, transcriptomics, and bioinformatics basis of breeding strategy. This is what we developed. To go for it, so there was a gap. The gap was we have uncovered the missing link with respect to the sex-specific and maternal inheritance. It is not it. The fact is that higher quantum of HSP90 and 70 genes transcripts was obvious in male and female larvae, and they are going to express differently in different instances. The question is whether you have to look for first instance, second instance, third instance, or fourth and fifth instance. The question was, most of the scientists, when they are developing thermotolerant breeds, they're focusing only on fifth instance, exposing them to the 35 degrees centigrade for 6 hours. Then that was the question to me. What about the first instance, second instance? All instance contribute together to make the larvae better and successfully spin the bucket. So then, based on this information what I acquired, then we start analyzing. So the female appears to be more tolerant to the thermal stress than males. Therefore, the female pure Mysore being used to grasp with the male CSR2 by 1 to bring it together, both tolerance as well as the productivity. So then we started thinking of towards this direction. Then we developed one hypothesis. The molecular chaperone activity of HSP90 and 70 should be understood very well, wherein it is going to protect the cellular proteins from denaturing during acute stress. But the question is not yet solved. Why? Because, as I said, in the silkworm body, there is a cellular machinery. As we see that central dogma is working very well in the body. So when cellular machinery is functioning in synthesis of HSP protein, is it possible concurrently to synthesize the silk protein? So therefore, the cellular machinery has to stop the synthesis of its protein, and then it has to switch over to synthesis of silk protein. So therefore, we need to be balanced that. So we have to make the cellular machinery to work in such a way and switch over from one protein synthesis to the another protein synthesis. So therefore, we have to look whether the cellular machinery allow the cellular machinery to become functional in biosynthesis of silk protein on cessation of thermospace. This is what we look at. So if you really look here, so HSP expression is not alike in both the sexes, which is in fact preferentially regulated in male and female larvae. So then we started using some biometry tool where we could able to understand the interaction of HSP90, HSP70, and other stress protein. So we came to know that HSP70 and HSP90 both coordinated or work together to support the growth and development of the silkworm larvae as well as the silk protein synthesis. However, it is going to protect the cellular damage due to the thermal stress. And then it has to switch over for the silk protein. And once we develop this concept, then start applying to that. So please change the strategy now. So far, we were discussing, we were talking about, we were thinking about only exposure of the silkworm larvae, either external or larval stage, with only 30, 45, 45, and studying only the shock response, but we never thought of their survivability or survival. But surprisingly, one fine day, when we expose this silkworm larvae or a silkworm eggs to the heat shock temperature of 45 degrees centigrade, fortunately or unfortunately, we could be able to get only small number of larvae of different cells. So generally what you do when the hatching is does, when you get the different size of the larvae, we throw them because it is not equal or uniform. Then we just took a chance, okay, let us rear all these silkworm larvae, how they're going to respond. At the end, eventually, we could be able to get some of the larvae grew better than the normal larvae, what they were hatched from the normal egg. Then some of the larvae are weak, some of the larvae are better. So when we get this diversity, then we thought of something is going on there. Okay, let us explore it. Then we stop thinking of conventional heat shock expression of heat shock, response and everything. Then we start looking at how it is going to be work together. So instead of using the cubids, then we start switching over to the hybrids as well. In that context, we could be able to get only the smaller size cocoons and the larger size. See the difference of these cocoons. The one is producing the smaller cocoon, another one is producing the longer cocoons, where the larva is healthy, and eventually we could be able to get the mark. Unfortunately, we got only one mark. Then we are desperate like anything. But we never lost our interest. Then we extended this study towards larva. We exposed the silkworm larvae to 45 degrees centigrade again. So far, we are talking about only the 40 and 35 degrees centigrade. Conduct all this heat shock protein expression, single dimensional electrophoresis, two dimensional electrophoresis, RT-PCR, then put up the paper publication and end up. That's what we used to do that. But one fine day, with curiosity, as one of my students, okay, let us see what is going to happen if we expose to 45 degrees centigrade. When you expose 45 degrees centigrade, that is the fluid, that is CSO2, you could be able to get only 2 larvae. Out of this, out of nearly 30 to 40 larvae we exposed to 45 degrees centigrade, heat shock for 2 hours and the recovery period of 2 hours, we got only 2 masks. Imagine, when I got these 2 masks, I was so happy, but unfortunately, these two masks were female. So my story is just lost. Then I was literally worried about it. There are only 2 female masks which are derived from 45 degrees centigrade. Then there are no male masks. Then how can you go for that? Then I asked my student, okay, let us repeat this experiment. So then We never stopped this experimental approach. Then we started using, instead of single hybrid, we started using hybrids. Then when we exposed the FC2 silkworm breed to 45 degrees centigrade, it has given a different results than that of the pure breeds. It was surprise to me when the pure breeds are giving a different results, when the hybrid is giving two different results, then I started analyzing these genome sequences or genome informations. So wherein the hybrid has given the mixture of the genome, where the pure breed has homogeneous. Then I exposed the silkworm larvae to 45 degrees centigrade heat shock for a period of 2 hours. Surprisingly, I could able to get 20 larvae from each batch of application. Then we never leave even a single larvae. We nurtured the single larvae separately. Then we started growing. Then, though there is only one larvae used to grow, there are 2 larvae used to grow, there are 10 larvae used to grow. Then we used to nurture all the single larvae to reach the final state to spin the cocoon. So likewise, we could able to complete 12th generation. That what we call as multi-generational heat shock response. We made it we undergone multi-generation heat shock procedure or strategy for a period of 12th generation. At the end of the 12th generation, you could able to see there are 6 new silkworm lines that we have successfully developed. We say that the line 2, line 4, line 5, line 9, 10A and 10B. Of course, initially we got nearly 20 lines. After subsequent screening and everything, some of the strains or breeds were lost, some of them are survived. But during the course of evaluation, we might lose some of the potential breed as well because of our ignorance. But if you people start doing this, try to look into each and every step where you could be able to get any screen. So all these 6 silkworm lines, what we develop, they are all fixed their original characters. So therefore, you could be able to see LBL9, LBL5, both of them performing better. Then, after developing the new silkworm lines here, the next question is, shall we take it to further the field, the field? So then, we subjected for this field evaluation. When I subjected all the silkworm lines for field evaluation, of course, it has given a good results wherein the NBL5 and NBL9 performing better, but NBL5 performing in all the season compared to that of the ruling silkworm breeds what we have now, CSR2 and CSR27. And we sent this paper for publication. Of course, it is published in Insect Journal of Biotechnology and Silkology. So having this substantial information wherein NBL5 is going better, then we thought of taking it to the farmer because ultimate goal is to enhance the livelihood of the humankind. So how we can able to enhance the livelihood of humankind is that if you really support the farmers to harvest a good quality cocoons and they get a good money and they will be happy. That was my strategy. Concurrently, when I have been doing all this job, so then the Chinese group also carried out this piece of work in China, and they could able to correlate this with that of the. They could able to get 4 different types of couples and started doing the subsequent work. So with this competition, especially whatever the silkworm breeds we develop, of course we say it's a good, but somebody has to be substantiated. So therefore, whatever the when I had interaction with the CSTR, Asian Technological Research Institution Bangalore, they are the pioneer in evolution of cocoon characteristic features. Then they asked me to submit some large quantum of the cocoons. Of course, I've given nearly substantial amount, 3 kg of each breed cocoons to them. Then they subjected all these breeds, whatever I developed. Of course, I've given only NBH1, NBH2, NBH3 with a single hybrid. Then they have really gave the certificate. They really says that. So these breeds could able to produce 3A to 4A grades of cocoons. So that is supported me to go further. So these are the characteristic features. Of course, you could see that NBH1, NBH2, NBH3. These are the three single hybrid classes. We have developed 6 new silicon lines here, six new silicon lines. So these 6 new silicon lines are crossed with another biotin grid, that's CSR2, and the resultant one is NBH1, NBH2. So the NBH1 substantially has given a good coupling weight, shell weight on O, and then see, these are the coupling characteristics of NBH1, NBH2, NBH3. These are the couples we sent for CSRDI, and then they would analyze the coupling characteristics features, and they said that these couples can aid to yield 384 red seals. So with these promising results and their advice or support, then we could able to produce large quantum of silicon lanes. So then I had requested one of the private grainier, wherein the private grainier produced NBH1. When it is see that, we could see the fecundity is higher. In case of the normal CSR2, it is about 550. But now we could be able to have 675 number of eggs per lanes. And then hatching percentage is 98. Then DFLS produced for the benefit of the farmers is 479. And cocoon harvested by the farmers is 449. And during that time, the cocoon rate, the cocoon sold was 360 rupees. Now the cocoons are going given for 1,000 rupees. To say that this NBH1 cocoons will produce in our laboratory by making use of the help of the MSc students and their skill development training program, we sold these cocoons for the price of 2,000 rupees. 2,000 rupees per kg of the cocoon. So this is how we thought, yes, these breeds can potentially go to the farmers. So then they prepared the silicon disease-free lanes at a larger quantity. Then these are the fecundity of new silicon breeds. And then with the help of the students, with the help of the Kerala Jockey Center, Mr. Jagdish, then we have produced the jockey worms in the support of Kerala Jockey Center in Maladi. So all this jockey which we produce, crashing with NBH1 with the ruling FC1, NBH1 with ruling FC1, then FC1 into FC2 is the ruling one, FC2 into FC1. So then it is giving a good result. All these lanes were distributed to the farmers. So our main intention is to distribute these lanes to the farmers. So thereby they could be able to harvest the good quality couples. And these are my students and these are the farmers who has taken these new breeds at the field. So eventually they could be able to get this is how our students with the farmers, they used to work together. These are the tracking center where our students work there. And then these are the cocoons and the larvae retrieved from the farmers. So ultimately, what I could be able to say here is that we could be able to see the comparative statement. The FC1 and NBH1, the larval weight is 5.9 grams. Whereas in case of CSR2 and FC1, FC2, which is conventional breed, is 5.6. Then the shell weight is also increasing. Ultimately, you could be able to see the filament length of FC1 into NBH1, our blue breed, yielding 1,185 meters of silk, where in case of the existing, the FC1, FC2 double hybrid is giving you only 886. There is a difference of nearly 300 meters of silk per cocoon. Or see, this is how the silk productivity is enhanced with the new silicon breeds. So thereby, we could be able to substantially supporting the farmers with this new breed, and that's how the built-up protein expression in terms of its response now converted into technology. That technology is utilized for production of the new silicon lines, single hybrids and double hybrids, and these double hybrids, single hybrids are distributed to the farmers, and those farmers are happy and they are fetching the good price by harvesting good quality couples. And this is how the field shop technology is involved in announcing human livelihood. So this is all about my presentation. Thank you. Hello? Hello?
Yes, sir.
Are there somebody? Yeah, please.
Hello, yes, sir.
Hello? Can you hear me?
Yes, sir. Yes,
Yes,
sir.
sir, we are there.
Yeah.
Yes.
Yeah, please. I'm through with my presentation. If you have any questions, you are free to ask me.
Hello, sir.
Yes, please.
Sir, like whatever that strains you have developed and given to the farmers, do
Yeah.
they work do they work in?
Yeah. Sorry, I can't hear you.
So please
Like,
unmute.
Those
Dr.
strains.
Lakshmikant, please unmute your
Yes, sir.
speaker.
I have unmuted. I have unmuted, sir. I have unmuted.
Okay. You can speak now. It is audible.
Yes, sir. So whatever the genotypes are, can it be grown in every geographical conditions throughout across India?
So now we are testing it in Karnataka. We are asking the permission from the central board as well as the state government to extend it to the further.
Okay.
We need to go
Currently
for a different. Yeah.
Okay, sir. Okay.
Currently, we are located in Mysore. We are testing all these breeds in Karnataka, particularly in Madhavali, Mandya area.
Okay. Thank you, sir.
Yeah.
Hello, sir.
Yes, please.
Sir, this is Kavya from Central University of Tamil Nadu.
Please,
sir, you generated multi-generation heat shock line for 12 generations, I remember. So, sir, in every generation you gave the heat shock, or you gave the heat shock and then were doing back cross every time?
No, madam. Especially what we did is that we have given the heat shock, we have developed a strategy. We have given the heat shock every generation until 4th generation. Okay.
Okay, sir.
And then, as per the scientific suggestions, we have, without heat shock, we regenerated them to recover the loss of recovery vigor. And then we continued the heat shock again in the 5th, 6th, 7th, another 4th generation with the same trend, with the same strategy. Again, we have we have not given the heat shock in one generation. Again, to revive them, to retrieve their vigor, and then we continued another 4th generation with heat shock. So ultimately, we reached 12th generation, wherein all the characters are fixed. During the course of this, we have subjected a stringent selection with respect to the cocoon characteristics, with respect to the pupal characteristics, with respect to the servability. Then every generation is to follow a stringent selections, and we classified those different cocoons into different lines. And then we started analyzing all these different lines separately with the proper replication and all under fluctuated environmental conditions. For this, we are not maintaining any congealed environmental like 25 plus or minus 1 degree centigrade. No, because if you maintain those breeds in 25 plus or minus, it's a congenial, and they're not going to express their, they cannot able to express themselves. So therefore, we have given the fluctuated environment. Because of these fluctuated environmental conditions, where the heat shock protein get expressed, and then acquire that tolerance level, therefore they could able to overcome this heat stress further, and that's a new breeds.
Okay, thank you, sir.
Yes,
sir, one more question.
Yes,
sir, both HSP 70 and 90 are indicated in tumor genesis, right? If I'm not wrong.
Yes.
So, like, how can you correlate this better growth and better cocoon, better quality cocoon to a gene that is indicated in tumor genesis?
Yeah,
like, what is your comment on it, sir?
Yes, madam, it is a good question what you raised it. For that, every generation, first of all, we used to run electrophoretic gel. Whether both of genes are expressing, every generation, every stage, first and second stage, we used to run this gel. Then we used to see whether in that batch the protein is expressing or not. If the protein is not expressing, we used to define them as a separate line. If the protein is expressing, we define them as a separate line. Then we enter for RT-PCR as well. So when we see that RT-PCR, of course, I have not presented almost all those data because it's a huge data. Then if at all exclusive the only that RT-PCR, PCR, I can able to present all the detailed information. We confirmed with RT-PCR as well. Surprisingly, you know that in Q breed, the RT-PCR results is different, and our breed RT-PCR different is different. Surprisingly, the Q breeds, whatever the CSRT is there, that RT-PCR upregulation is more. In our breeds, the upregulation, rather than upregulation, we have a downregulation. And we are under the, again, challenge, why these differences is occur. And we are now looking at, you know, now still we are going, whether there is genome organization. For that, we are taking the genomic DNA of these new lines and designing the primers, see the polymorphism, and see the SNPs variations. That work is still going on in our lab. One of the PhD student is working on that line, extract the genomic DNA, does the PCR, then use the PCR product for variations again. So we are trying to look into this gene diversity rather than genome diversity.
Okay. Okay, sir.
The next step is, next step is we are looking is gene diversity, but not genome diversity. I'm looking the diversity within the gene, within the HSP 17 HSP. I'm looking at now.
Okay.
Yes.
Thank you, sir.
Very good morning, sir. I'm Dr. Kalishwaran from Tamil Nadu, sir.
Please tell me, sir.
Sir, I'm Kalishwaran from Tamil Nadu, sir.
Yes, sir. I'm listening to you. Please tell me, sir.
Sir, in my research work in the fish immunology, that the time also I studied the HSP.
Good.
Even a normal stress condition, HSP has been increased. The expression is more or depends on the conditions.
Sir, especially the response of this organism to express the HSP protein, it is above the threshold level. In normal condition, HSP expression is there to maintain homeostasis. It has to be there. Because I have shown one slide, is it? Western blot slide I have shown you. Even in normal larvae, HSP expression is there. But when we expose to heat shock, to overcome the tolerance, the rate of expression is more here. That will measure in terms of 1-fold, 2-fold, 3-fold. Higher the rate of expression, more tolerance level. That's what people say that. But in silkworm, if the higher rate of expression HSP, it is not synthesized the silk protein. It is negatively correlated. So we have to balance between the HSP expression to acquire the tolerance and the production of silk as well. This is the limitation with silk. But it may be different in case of different organisms.
Okay, sir.
Did I answer you?
Yes, sir.
Thank you.
Thank you, sir.